Hello, welcome to my presentation on CRISPR. Hello, my name is Shanna DeNovo, and this is my presentation on CRISPR. So in order for the presentation to start, we're going to go over some objectives. So the objective of this presentation is that the students will learn some basic terminology related to biotechnology, the history of CRISPR, like what CRISPR is, how the CRISPR-Cas9 system functions to edit genes, and how CRISPR is currently being used to manipulate genes in the lab and in the real world. Okay, so at this time there's a little introduction activity to get the kids interested and thinking about the effects of gene editing. So in 2 minutes they're going to discuss this with their partner. So they're going to imagine if they've been offered a deal from a genomics company, they can get a free genome sequencing and analysis of all of their DNA that includes a report of their ancestry, traits, and medical profile. The medical profile tells about diseases for which they have a low risk of getting and also those that they have a high risk of getting. And the students need to answer if they're interested and why or why not they're interested. So this activity is just to really get them thinking about the repercussions and implications of getting their genome going. So students will get a minute for each partner to kind of discuss what they think. Okay, key terms. So key terms are genetic engineering, and what genetic engineering is it's a process of manipulating genes and genomes. So genetically changing those genes and changing the genome. And then biotechnology is a process of manipulating organisms or their components for the purpose of making useful products. So biotechnology is a new and exciting field, and there's so much to learn and still so much to discover. Okay, some tools that genetic engineers use are restriction enzymes. And we learned about enzymes previously. And remember, enzymes are proteins. So the restriction enzymes, also called restriction endonucleases, are used to cut the strands of DNA at specific regions that we call restriction sites. So they can be programmed to cut a specific place in the DNA. And the restriction fragments are the result of those cuts. So restriction fragments have at least one what we call sticky end, or end that isn't parallel to the other end. So that's a single-stranded end that sticks out, that can bond to other things, and that's why we call it sticky. So then we also have DNA ligase, which we've learned about in our transcription translation unit, and that is what joins the DNA fragments together. Okay, here we have a CRISPR video. So we can go ahead and click on the video. At this time, go ahead and just pause the recording and watch the introduction video on CRISPR. For the purposes of this, I'm not going to show the whole video. Okay, so moving on past the video. So the video is a great introduction and gives kind of a visual of CRISPR and why we have CRISPR. So the big question, where did CRISPR come from? So this great technology developed, where did it come from? So CRISPR are repetitive DNA sequences that are commonly found in over 40% of bacteria, prokaryotes, and archaea. CRISPR, together with what we call CRISPR-Cas or CRISPR-associated proteins, it's part of the prokaryote defense system against viruses. So just like we have an immune system, prokaryotic cells also need an immune system. So this CRISPR-Cas system protects the cell by allowing DNA sequences that are identical to past pathogens into their genome, so if they get invaded by those pathogens again, they can recognize them as foreign and attack. So RNA, called CRISPR RNA or cRRNA, is transcribed from the spacer sequences at the CRISPR locus. We'll look at a picture of this next. The cRRNA directs the Cas proteins in the bacteria or archaea to identify the foreign invading sequences and cleave or cut it so that it's able to kill that foreign invader. So this is an adaptive immunity mechanism that is in the bacteria and archaea. It's evolved as part of a defense system, so like a defense against foreign genetic material like phages. So there's several different types of CRISPR pathways in bacteria and archaea. The common type that they use for genetic engineering is the type 2 CRISPR-Cas9, and that's because it creates a double-strand break in the targeted DNA, and it also is more simple and only involves 2 proteins. Okay, so here is our picture that I was mentioned earlier, and it goes through the different stages. So stage 1, the foreign DNA is added to the genome. In stage 2, the RNA is processed, and then if you look over to stage 3, the RNA is guided. The RNA guide targets the viral element. So it goes in, the RNA guide can target that viral element, that virus that you see invading right there in the upper screen. Can you see my pointer? I don't know. And it can cut it, and then it kills the virus so the virus can't infect the bacteria. Yay, prokaryotes. Okay. Trying to move on here. Okay, so what is CRISPR? So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. So it's a genome editing technique that's able to target a specific section of DNA. And then at that section, with the guide RNA, it makes a precise cut or break in the target site. And some of the awesome things it can do is make genes non-functional. So it stops the DNA, making the genes non-functional, or replace one version of a gene with another. So that could be great. If there's a problem, you can replace the problem with a solution. So how does this work? So when we use CRISPR as an experimental tool for genome editing, it has 2 main components. The first component is a targeting system that finds the right place in the genome to cut. This is achieved by a molecule called guide RNA, or gRNA, which has the same genetic sequence as the target genomic site. The second component for making is a component that actually makes the cut to the DNA. This consists of a DNA cutting enzyme called the nuclease, and in this case it's called Cas9. So that's why we call it CRISPR-Cas9. When both of these components are delivered into a cell, the gRNA will bind to the target genomic site through a complementary base pairing, meaning A's with T's and G's will bind to C's. In the process, the guide RNA helps bring Cas9 to the target site to make a cut to the DNA double helix. The cell's natural DNA repair mechanism will close this gap, but because the process is not perfect, a few DNA bases will be added or deleted. This renders the original gene, a gene variant linked to cancer or related to HIV infection, non-functional. Yay! So kind of cuts that gene out, and now it doesn't work, and this could be good if the gene is not a positive gene. So CRISPR can also be used to replace an undesired version of a gene, like one that causes the disease, with a desired copy. In this case, a desired version of the target gene can be placed into the cell along with the gRNA and Cas9. The cell will then use this alternate sequence as a template to repair the broken DNA through the process of homologous recombination, or copying the new sequence into the genome. Okay, so the ways that CRISPR or the body can actually repair itself, or the cell repairs, is either the non-homologous end joining, NHEJ, or homologous recombination. So scientists have been able to use the cell's natural ways of repairing to kind of insert the new gene that they want. So here's a picture of the two possible pathways. So the NHEJ on the left there creates those knockouts. It's a little more error prone, which is not always good. So, and occurs with higher frequency. So the other pathway is the HR pathway that I mentioned previously, or the homologous recombination pathway. And this requires a homologous template donor. So if they want to do a knock-in to put a template in, this is the pathway that they use. It's not very efficient, but it's more accurate in terms of inserting a new gene and kind of fixing a problem. So why do we care about CRISPR anyway? Well, CRISPR is a process where researchers introduce modifications into genes. So it can result in disruption, insertion, or replacement at any location that they desired. So it can control gene expression, which is exciting. And it's great because it's simpler and easier to use than other previous genome editing technologies. And it basically brings back the possibility to have gene therapy and edit genes to fix diseases. Okay, so I mentioned gene therapy. What exactly is gene therapy? So using genetic technology to directly treat genetic causes of diseases is called gene therapy. It's kind of been a long-standing aspiration for physicians and scientists and patients. Some diseases like cystic fibrosis or sickle cell anemia are relatively well understood and can be caused by variants in just a single gene. If these disease-causing genes can be corrected or replaced, then the hope is that we could cure the disease or prevent it from worsening. However, it's more difficult or more complex conditions such as heart disease, diabetes, many forms of cancer, which are many genes kind of interplaying together that create that negative environment. So it could be more complicated in cases like that. But in the case that you see in the picture here, you see the picture of sickle cell anemia and cystic fibrosis. This is just one gene causing these problems. So cystic fibrosis is a genetic disorder where thick, sticky mucus in the respiratory pathways lead to breathing problems, developmental delays, and infections. And then cystic fibrosis is caused by mutations in one gene called the CFTR. Treatments have been very hard to find, and cystic fibrosis has been one of the most anticipated targets for gene therapy. One of the main challenges for gene therapy is the ability to safely and effectively make the desired genetic changes to all the cells, because we have so many that need the gene to function. In the case of cystic fibrosis, all of the cells in the lungs and sweat glands may need functional CFTR genes in order to properly produce mucus and other secretions. While no therapies have yet been approved for the use, in recent years there have been some promising experimental results in mice and pigs where the majority of their symptoms have been reversed. Okay, on to sickle cell disease. So sickle cell disease is a genetic condition characterized by mutations in the oxygen-carrying proteins of the red blood cells. So sickle cell disease causes hemoglobin proteins to stick together and lead to the red blood cells turning into sickle cell shape, like is pictured here. There is also another promising approach which could take advantage of a secondary hemoglobin gene that functions in fetuses and then gets turned off shortly after we are born. This hemoglobin gene, called the fetal hemoglobin gene, is not affected by mutations that cause SCD. So one potential gene therapy would treat SCD by turning on the fetal hemoglobin gene and turning off the adult version. Experiments using this approach seem to work well in mice, and clinical trials in humans are likely to begin soon.